All right, so if you're on the podcast and you're just joining us, make sure you get one of my handouts from somewhere. I'll have to send you guys the link. I have all these uploaded into Google Drive, and I'll just send you the link to that hopefully this week. And it's got the mock exams and everything we'll be doing for the rest of the try. So we're going to be starting on Chapter 5. There is no handout for Chapter 5, so please don't go looking for it. What we're dealing with in this chapter is eicosanoids. And any questions, first of all? Happy thoughts? A little bit of smiles? It's only try 2. It's no big deal in the long run. Yeah. All right, so we're dealing with eicosanoids. Eicosanoids are inflammatory mediators. So these guys are involved in inflammatory processes, blood clotting, vasoconstriction, vasodilation, all that stuff. That's what we're going to be talking about this chapter, is how these processes, how these specific things, these eicosanoids, these molecules, are involved in that process. So first of all, they are local hormones. So some old terminology. You can TQ this right now if you want. It's going to show up a couple times throughout this chapter. That their half-life is from seconds to minutes. They are very, very, very short-lived. Very short-lived. We make them, we use them, they're gone. They don't stick around long. You don't want these guys mediating inflammation minutes, hours, days after the fact. They act in two ways. They're local hormones. They act in both a paracrine fashion and an autocrine fashion. What is our third method of action? Paracrine, autocrine, and bloodstream, endocrine. They are not endocrine hormones. They don't last long enough. They're seconds to minutes, which means by the time they would flow through the blood out to somewhere else, they would be not an eicosanoid anymore. They would have been degraded. So they deal in a local fashion. You need to know these two things. We talked about them in Try 1. I'm sure you talked about them before. Start with autocrine signaling. Autocrine signaling means the cells can signal other like cells. If it is cell A, it can talk to itself. It can activate itself. Some auto activation, or it can activate other A cells. You can think of your own analogy for something that works on other things of its own type, but I'm going to leave your dirty minds to that. Paracrine cells doesn't care who it acts on. As long as it has a receptor, it's not just acting on A cells. It can act on A cells, it can act on B cells. It acts on other cells. Remember, these are both local, not endocrine, both local, but specifically autocrine is auto, it's self. Wow, never had that happen. It's self-activating because it only acts on self cells. Not just itself, but other cells of its like. Questions on those? Pretty easy concept. Okay. Here's the overview of what we're going to be looking at this chapter. So eicosanoids mediate lots of different stuff. We've got a few on here starred that you might want to know. We're going to kind of talk about all of them. The big thing is they mediate the inflammatory response. That's the primary goal of eicosanoids, is to mediate the inflammatory response. They produce pain and fever. You can write in right here if you want, prostaglandins. We're going to talk about 4 or 5 classes of eicosanoids, and this is one of them. Prostaglandins are responsible for pain and fever. So if you see pain and fever, you pick prostaglandins. They regulate blood pressure. If you want to change this, it should say vasoconstriction, vasodilation. I don't know if that's fixed in his book, but it was not in the lecture manual, so make sure you change that if it's not. They induce blood clotting. The blood clotters are our thromboxanes. If you see blood clotting, you pick thromboxanes, flat out. They control several reproductive functions. If you see reproductive, smooth muscle, uterus, that's prostaglandins as well. Not such a huge point, but it is kind of a minor one. Inflammation, pain, fever, blood clotting, those are our primary ones for now. Water and salt, sleep, wake, etc., etc. Not a big deal for the rest of them. So that's just a general overview. You're going to have to know the details about each one of these if you didn't get that fact from the fact that I put prostaglandins and thromboxanes in that list. When we go through each one, you're going to have a little list that you're going to know that prostaglandin DIE do this, 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 and this, and prostaglandins I do this, this, and this. So you'll have to know those specifically, but that was kind of the overview. So down here, they are fatty acid derivatives. We've just been studying fatty acids, so now we're going to a derivative of fatty acid. All eicosanoids are derived from arachidonic acid, TQ, TQ, TQ. All eicosanoids are derived from arachidonic acid, which is a 20-carbon fatty acid. Arachidonic acid is one of your nutritionally essential amino acids. It was in that list I gave you way back when. 20 carbons, 4 unsaturations. That's what the colon 4 means. It is the precursor of all eicosanoids, etc., etc. Now down here, this is also important. We cannot synthesize arachidonic acid. It is dietarily ingested. It comes from linoleic acid, aka omega-6. Comes from linoleic acid or linoleate, if he puts the conjugated base version. Oh, right there, or linoleate, which is an essential fatty acid from plants. So we take this oil in, although primarily you get arachidonic acid from animal products, but since it's a plant oil, how do we get it from animal products? Since it's a plant oil that we're deriving arachidonic acid from linoleic acid, but we primarily dietarily get it through meats. Why? Because cows eat plants, because animals eat plants. So they build up a lot of arachidonic acid. So if you eat plants, you don't actually get a ton of arachidonic acid in you. Oil is going to be a little more concentrated because it's the fat itself, so you're going to concentrate it a little. But primarily our main sources of arachidonic acid are meats, especially red meats. Because same thing, as you move up the food chain, this stuff just gets concentrated in there. So arachidonic acid always comes from linoleic acid. Arachidonic acid is the precursor to all eicosanoids. And linoleic acid, I said that, dietary linoleic acid. We can't synthesize it in the body. All right. This is kind of a key point, or an interesting point. We're going to kind of hit it into the ground so you can do what you want with it. Arachidonic acid is esterified with phosphoglycerates and produces glycerophospholipids. Anyone want to interpret that for me? Arachidonic acid is esterified with phosphoglycerates and produces glycerophospholipids. It's a fatty acid. We're making a phospholipid out of it, which means that to store this arachidonic acid, we attach it to a glycerol backbone with a phosphate at position 3. We make a phospholipid and we dump it in the cell membrane. So we store our arachidonic acid in all of our cell membranes, so that when we need arachidonic acid for a local inflammatory process, all we have to do is go to the cell membrane and rip some out. So it's stored as a phospholipid in the cell membrane, which is going to make a lot more sense when we talk about this. So you guys remember these from chapter 1. We want to break down a phospholipid. A phospholipid is a fat. To break down a phospholipid, we have 4 enzymes that break it down. Phospholipase, A1, A2, C, and D. Remember those? So if this guy's stored as a phospholipid in the cell membrane and we want to break this guy down, we need to use one of those phospholipases. There's specifically 2 pathways in which we do this. We can use phospholipase A2. Where does phospholipase A2 break something off of a phospholipid? Second carbon, position 2 carbon. So if we have arachidonic acid stored on the second carbon or on the first and second carbon and we want to free this guy at the second carbon, we use phospholipase A2, breaks it off the second carbon. Now we do not use phospholipase A1 on this guy. We don't want to do that. So one of our options is just to free a single arachidonic acid from a phospholipid. Whether or not it has 2, we don't care. We're just taking the one from position 2. The other option is this guy has 2 arachidonic acids. We want both arachidonic acids. So we come in here and we use phospholipase C. And we cut off the phosphate and the head group. Now we have a diacylglycerol. That diacylglycerol has 2 arachidonic acids on it. Then we come in, once we have a diacylglycerol, and we use phospholipase A2 and get 1 free one, phospholipase A1, get another free one, and now we just have a glycerol, 2 free arachidonic acids, and the phosphate group floating around with the head. So what you need to think about is, if you use phospholipase C, you get 1 arachidonic acid. You take it from if you use phospholipase A2, you get 1 arachidonic acid, you take it from position 2. If you want both arachidonic acids, and it has both arachidonic acids, you first cut off the head group, and then you cut off each of these one at a time. That's what this next couple pages is talking about. So phospholipase A2 hydrolyzes the acyl groups at the second position. I would know that, just like before. Phospholipase C hydrolyzes the phosphatidylinositol head to form diacylglycerol and phosphoinositol, which is the head group plus the phosphate. And then we have a diacylglycerol lipase and a monoacylglycerol lipase as we cleave those other arachidonics off. So over here, ignore this top part real quick, we have 2 random fatty acids up here that have arachidonic acid attached to them. We're going to assume this guy has 1, phosphatidylcholine. We use phospholipase A2, we get 1 free arachidonic acid. From position, phospholipase A2 takes it from position what? Position 2. Good. Over here we have we have a phosphatidyl something with lots of arachidonic acid on it. Both of them. We want both, so we use phospholipase C. We get rid of that head group and the phosphate group, and all we have left is a diacylglycerol. Then we use diacylglycerol lipase, or one of your phospholipases, and you get a free arachidonic acid and a free monoacylglycerol. And use monoacylglycerol lipase, and you get a free arachidonic acid and a free glycerol molecule. You could call those DAG and MAG if you want to, diacylglycerol, monoacylglycerol, and therefore DAG lipase and MAG lipase. But do you guys get the concept? You're going through this chain because you're cutting off the head group and all you have left is a diacylglycerol. How do you cut a diacylglycerol? You use a diacylglycerol lipase. Questions on that? Got to know those. Those are your ways you free it. So if he tells you you have a phospholipid with a 20-carbon fatty acid at position 2 and an 18-carbon fatty acid at position 1, and you want to get a free arachidonic acid. How do you do it? Phospholipase A2, because that position 2, at position 2. 18 carbons at position 1, 20 carbons at position 2. You want a 20-carbon free arachidonic acid. 18 carbon is what? Stearic acid. Or oleic acid. All the omegas are 18 carbon, so you're right. Okay, now up here at the top, big point. Big point. Activation of phospholipase A2 and C occurs when histamines and cytokines are released. What have you learned histamines and cytokines are released during? Allergic reactions, tissue damage, all of that stuff causes cell damage, causes the release of histamines and cytokines, which are inflammatory mediators, and this is that pathway. So you haven't learned the inflammation pathway yet, because now we're going to learn it. So something happens to the cells. They release histamines and cytokines, whether it's an allergic reaction causing inflammation or tissue damage causing inflammation. But histamines and cytokines are released, and they activate the phospholipases. That's all they do. Histamines and cytokines, I'm sure they have a few other effects, but they activate phospholipases. Phospholipase is free arachidonic acid. Arachidonic acid is going to go through the pathways we're going to talk about and become these inflammatory mediators, each of which has their own effect to cause inflammation or clotting or vasoconstriction, vasodilation, et cetera, et cetera. So you see where we're kind of going with this? Histamines and cytokines bind to their specific membrane receptors, and I wrote over here, activate the phospholipases, either A2 or C. That's what they do. Down here, you've learned this before, or not really learned this, you've heard this before. When someone has consistent pain and inflammation, they get a cortisol or a prednisone shot. They get a corticosteroid shot locally, which decreases their pain and decreases their inflammation. And remember, prostaglandins, which are one of our eicosanoids, are responsible for pain. And eicosanoids are responsible for inflammation, which we're still talking about. So if we want to decrease this pathway, all we have to do is shut down the production of free arachidonic acid. How do we do that? These corticosteroids, which are used as anti-inflammatories, inhibit A2 and C. I think A2 and C, it just says A2. But they inhibit phospholipases. They stop free arachidonic acid from being produced. And if you don't have free arachidonic acid, you cannot produce eicosanoids, you don't have pain, you don't have inflammation locally due to this pathway. So histamines and cytokines activate the phospholipases. Corticosteroids inactivate the phospholipases. Make sure you know those guys. Okay. Now, on this page, on page number 54, for those playing the home game, we've got 4 mechanisms of action of glucocorticoids. Glucocorticoids are a type of cortisol, a type of steroid, so it's one of those things that we said is going to inactivate the phospholipases. So, there's 4. You really need to know 3 of them because one of them is kind of repetitive. So we're going to start at the bottom. Corticosteroids inhibit phospholipase A2 by inducing the synthesis of lipocordins that block PLA2 activities. They stop phospholipase A2. If they stop phospholipase A2, you get no free arachidonic acid, you get no eicosanoids, you get no inflammation, no pain. They suppress the transcription and translation of COX-2 enzyme. We're going to talk about that guy. COX-2 produces prostaglandins and thromboxanes. If you can't produce prostaglandins or thromboxanes, no inflammation, no pain. So corticosteroids stops the free arachidonic acid production, stop the production of the enzyme that makes the eicosanoids that we care about, and they stop the secretion of chemotactic factors. What's a chemotactic factor? Cells and tissues. Micro. What is chemotaxis? Chemical taxis. When you have a chemotactic factor, it's something that is secreted locally that creates a chemical gradient that draws white blood cells and other mediators of repair to it. So if you have cell damage, whatever's happening there is going to secrete chemotactic factors, which calls all the white blood cells and all the repair cells over and says, hey, we got an issue. We need some cleanup, we need some damage control, we need some repair. So they stop the process of bringing stuff to the area to repair it. They stop inflammation, they stop pain. Now the reason I said you can kind of ignore this one is because they inhibit or limit the recruitment of leukocytes, monocytes, macrophages. That's chemotaxis. The recruitment of leukocytes, monocytes, macrophages into the inflamed area is chemotaxis. So if you want a definition for chemotaxis, there's a very basic one right there. The local stuff will secrete chemotactic factors to draw white blood cells, macrophages in to clean up and repair that area. So glucocorticoids stop it by stopping the enzyme that frees arachidonic acid, the enzyme that makes eicosanoids from arachidonic acid, and stopping the bringing to the area of repair and other cells that are going to secrete more histamines and cytokines to get this process ramped up. All right, now we're going to start getting into the meat of this chapter. So any questions on what we're talking about, where we're going, what's for dinner, what kind of cookies I like, what kind of cookies you guys like? I like cookies. Lots of cookies. You don't have to bring me cookies. I'm just Jack Daniels cookies sound amazing. I wonder where I could get some Jack Daniels. All right. So first class, and I'm going to skip around in this chapter because I don't really like the organization. Dr. Sarkar, if you're listening, I'm sorry. We're going to talk about prostaglandins first. We're going to get all the way through with prostaglandins, then I'm going to come back and talk about the other boys. So prostaglandins are one of our eicosanoids. Like I said, we're going to be talking about 4 or 5 eicosanoids. They're one of our eicosanoid subgroups. They have a 5-carbon ring, which is a structural question you need to know. You need to know the structural characteristics of all of the eicosanoids. They have a 5-carbon ring, TQ, 5-carbon ring, originating from the chain produced by almost all mammalian cells except red blood cells. Does that seem important? Produced by everything except red blood cells. Make sure you know that. So they're drawn right over here. They regulate the synthesis of CAMP, and they stimulate the contraction of smooth muscle of the uterus. Remember I said that before, prostaglandins, uterine contraction. You haven't learned yet, prostaglandins, CAMP. Those are your big 2 right here. They also affect some blood flow. We're going to talk about that, some wake-sleep cycles and some other stuff, but these are your big 2. Now I want you to note over here, these are the 3 structural characteristics that you need to know for a prostaglandin, and they're going to be kind of similar to the other guys. They have a 5-carbon ring. They have a double bond between carbons 13 and 14, and they have an OH group at position 15. Please note that. All 3 of those are structural characteristics of a prostaglandin. 5-carbon ring, double bond between 13 and 14, and OH at 15. Hydroxyl group at 15. Those are the 3 key structural characteristics of a prostaglandin. If it loses any one of those, it is no longer a prostaglandin. So how do you think we're going to inactivate or degrade these guys if we want to? We're going to break the double bond. We're going to do something to the hydroxyl group. We're going to change the end. We're going to break the ring. If they don't have those 3 structural characteristics, they're not a prostaglandin. How long does it take to inactivate these guys once they're produced? Nice. Seconds to minutes. Okay, so I'm going to tell you this right now. They regulate the synthesis of CAMP. What receptor do they use? Nice. G-protein coupled receptors. Because these guys, and that'll be the very end of the chapter, they talk about these guys use G-protein coupled receptors. What else tells you they're what type of receptor is it? Is it a membrane or nuclear receptor? G-protein, is it a membrane or a nuclear receptor? Membrane bound. This guy's huge. He's 20 carbons. He's covered in negatives and positives and OH groups. He is a polar molecule. Polar molecules do not cross the membrane. Polar molecules use membrane-bound receptors. Since they use membrane-bound receptors, they use a G-protein coupled receptor. In this case, they regulate the synthesis of CAMP. Bam. Knowledge. So we're going to skip to page 56. the cyclooxygenase pathway, and I hope you guys are keeping up in your study materials, whatever you got in front of you. Right here, COX. The cyclooxygenase enzyme, aka COX, aka prostaglandin peroxide synthase, or PGH2 synthase. That's going to make a hell of a lot more sense here in a minute, so don't worry about the AKs. It's the COX enzyme. You guys have heard about this. You guys have heard about COX inhibitors, COX blockers. You guys have heard about COX blockers. There are two types of COX. You got to know this. Naturally occurring COX is COX-1. It is in the stomach. COX-1 is in the stomach. COX-1 produces mucus in the stomach. Inducible COX is COX-2. It's induced by inflammation and it produces our eicosanoids. COX-1 is in the stomach, active all the time, produces mucus. COX-2 is induced by inflammation, produced in every cell in the body except red blood cells, or induces inflammation, or induced by inflammation causing more inflammation, et cetera, et cetera. So, one of our primary COX blockers that you guys have heard about, what's a common anti-inflammatory? Aspirin, Tylenol, ibuprofen, meclofeminate, or whatever the, what? NSAIDs. Any NSAID, non-steroidal anti-inflammatory drug. It's an anti-inflammatory drug. Don't worry about writing this down. We're going to get there. They shut down COX. They are COX blockers. If they block COX, they stop COX-2, decreasing inflammation. That's what you want. You want an anti-inflammatory drug. What else do they shut down? COX-1. Because the first place they go is your stomach. They shut down natural COX-1. Because they're COX blockers. They don't care if it's COX-1 or COX-2. They shut down COX-1 in the stomach. COX-1 in the stomach produces mucus in the stomach. What's the function of mucus in the stomach? Protect the stomach from the acid. So what's the side effect of taking too many NSAIDs? Ulcers. Because you degrade the mucus lining of the stomach, you let the acid get to the stomach wall, you get ulcers. So it's because it's the same enzyme. Now they're coming out with COX-2 inhibitors specifically, which work a little better, but yeah. So you've got an overview up here at the top of page number 57. We've got arachidonic acid, which is what we're talking about. What is the precursor of arachidonic acid? What? No. Dietary linoleic acid, or linoleate, or omega-6. Good. Arachidonic acid has a couple of different pathways. We're not going to talk about the cytochrome P450 system. You'll deal with that in nutrition, or you might skip it. We're going to talk about the lipoxygenase pathway, which is going to produce leukotrienes and lipoxins. Easy. The L pathway produces your two L's. And the first one we're talking about is the COX pathway, produces prostaglandins and thromboxanes. So you have three fates of arachidonic acid when it's freed from the membrane. You have the COX pathway, producing prostaglandins, thromboxanes. You have the lipoxygenase pathway, producing leukotrienes and lipoxins, and you have the cytochrome system, which we're not going to be talking about. So, COX pathway produces prostaglandins and thromboxanes. Here's those structural TQs. Prostaglandins, fatty acids containing 20 carbons. I forgot to mention that. All eicosanoids are 20 carbons. A saturated 5-carbon ring, an OH at 15, and a double bond between 13 or 14. Please know the structural characteristics. They will show up on the test. And we're going to talk about a key difference between them. Prostaglandin synthesis involves the consumption of 2 molecular O2. You don't really need, well, you do need to know that in terms of it's an important point, but it's not really a clinical piece of information, aside from you can't make prostaglandins if you're oxygen deficient. But I'm going to assume that lacking oxygen is not going to care about prostaglandins at that point. But it is an important process that requires oxygen. That's why it's called cyclooxygenase. It's adding oxygen to it, the COX enzyme. First step is catalyzed by prostaglandin endoperoxide synthase. We have a nice little chart on the next page, so don't worry about it too much here. COX adds 2 molecules of O2. If we look on the next page, it's got all your little charts up here. Peroxidase activity converts the hydroperoxy group to hydroxyl group. Great. You do want to know this. To synthesize the first stable prostaglandin. The first stable prostaglandin is PGH2. PGH2 stands for prostaglandin H2. PGH2 can be reduced by any number of these enzymes. It can be converted, et cetera, which is what we're going to look at. Down here, you want to know this as well. PGH2 can be converted to PGI2. PGI2 is known as prostacyclin. It's produced in vascular endothelial cells. You got to know PGI2 is aka prostacyclin, and it's produced specifically by vascular endothelial cells. The other prostaglandins we're going to talk about don't have alternate names. They don't have specific production locations. That one you got to know, and we're going to go over him separately later. So if you look down here, we take our arachidonic acid, we cox it. Coxing it adds some oxygen, turns into PGG2. But we don't really care about PGG2 because PGG2 is not a stable intermediate. It goes right through a peroxidase enzyme and gets turned into PGH2, which is your first stable intermediate in the prostaglandin synthesis pathway. Now, the AKAs for this were prostaglandin peroxide synthase because this is a peroxide structure or an endoperoxide structure right there. So it's called prostaglandin peroxide synthase or endoperoxide synthase or PGH synthase because that's what it's doing. They're all synonyms for the COX pathway, which is really two enzymes right here. It goes straight from, well, it goes from arachidonic to PGH2 through a really flip-floppy intermediate. So arachidonic acid to PGH2 through a couple of different enzymes. You're likely going to get asked about the fact that it's called COX, cyclooxygenase. I don't think he's going to throw the other weird names at you, but I want you to be aware of those because boards might, and they might just show up on Sarkar's test. But basically, arachidonic acid consuming two O2s to PGH2, the first stable intermediate. Note that PGH2 has a 5-carbon ring, a double bond between 13 and 14, and an OH at position 15. Now, this is really hard. I know you guys are going to hate this. We can turn PGH2 as our first stable prostaglandin intermediate, but it's not used for a lot of stuff. It's an intermediate. We want to get to our final products. Our final products are PGD, PGE, PGI, aka prostacyclin, and PGDIE. Oh, that's it, I guess. And then TXA is not a prostaglandin. These enzymes are hard to remember. I know you're going to have to work to remember these. If we want to take PGH2 and make PGD, we use PGD synthase. If we want to take PGH2 and make PGE, we use PGE synthase. If we want to take PGI or PGH and make PGI, we use PGI synthase, and this one's hard. If we want to take PGH and produce thromboxanes, we use TXA synthase or thromboxane synthase. That's what it stands for. TXA is thromboxanes. So enzymes, no big deal. You guys don't even really have to worry about them. Just say what it is and say what it's making. Now, this is an important thing to note, that thromboxanes come from a prostaglandin intermediate. So the COX pathway produces both prostaglandins and the thromboxanes. And the thromboxanes come from a prostaglandin intermediate. We haven't talked about thromboxanes yet, but we will. Down here, I rewrote your little thingies right here. They produce, PGI is produced by vascular endothelial cells. We're going to talk about the fact that it causes vasodilation and inhibits platelet aggregation. You can write it down if you want. You don't have to. It's coming up in a typed up list, not in my sloppy handwriting. So that's your synthesis path. COX produces PGH2 from arachidonic acid, or PGH2 is turned into any number of these other guys right here. Now we're going to page 60. This is what your prostaglandins do. This is the chart you need to memorize, the little list. PGD, PGI, and PGE, easy way to remember this, PGDIE cause dilation. DIE cause dilation. I know it's spelled differently. I'm not a special person, but it helps you remember. PGD, I, and E cause dilation, which means they increase vasodilation, and they also increase camp levels. Why do they increase camp levels? Because they use G-protein coupled receptors. Perfect. Because they use G-protein coupled receptors, they increase camp levels, which is anything that uses a G-protein coupled receptor. Now, this list looks really, really scary. They decrease platelet aggregation, leukocyte aggregation, interleukin 1 and 2, T-cell proliferation, and lymphocyte migration. They decrease the immune function, or the repair function, whatever you want to call it. They decrease immunity and repair. And remember, we talked about that. Oh, actually, we didn't. Prostaglandin D, I, and E cause vasodilation and decrease immune function, decrease the activity of the white blood cells in that area. They decrease chemotaxis just like cortisol and prednisone and all the corticosteroids do. So what you need to remember about PGDINE is they cause vasodilation, DIE, dilation. They increase camp levels because they use G-protein coupled receptors, and they decrease immunity in general. Anything he asks you about immunity, it's decreased by Pg die, because Pg die makes you die. It decreases your immune function and dilates you. PGF is this little alternate. PGF basically does the opposite of Pg die. PGF increases vasoconstriction. It causes bronchoconstriction and smooth muscle contraction. There's your uterine smooth muscle contraction. Now, you also know that smooth muscle contraction must go with bronchoconstriction and vasoconstriction because those are smooth muscles, right? So if you're vasoconstricting, bronchoconstricting, you're smooth muscle contracting. Or if you memorize smooth muscle contraction, you know it's vasoconstriction, bronchoconstriction. So PGF does basically the opposite of die, causing smooth muscle contraction. That's prostaglandins. That's what they cause. You're going to learn later they cause pain too. I don't remember if we talked about it in here or what class you talked about it in. But prostaglandins are responsible for pain and fever as well, as he said in the beginning of the chapter. So make sure you know that. Maybe write that in somewhere. Prostaglandins in general cause pain and fever. Prostaglandins in general cause pain and fever. Specifically, di is this list and F is that list. Okay, we're going to skim back and look at some thromboxanes real quick. See where we're going. Thromboxanes, this is the light version right here on page number 55. Thromboxanes are a 6-member ring, not a 6-carbon ring, a 6-member ring, because one of theirs has one of their ring members is an oxygen. Do you think there's going to be a test question that says thromboxanes have a 5-carbon ring, a 5-member ring, a 6-carbon ring, or a 6-membered ring? Prostaglandins have a 5-carbon ring, a 5-member ring, a 6-carbon ring, a 6-membered ring.
This is the security department. Please be advised that the creek lot will close at 6 p.m. Please move your vehicle from the creek lot to the main campus at this time. Thank you.
So, 6-member ring, make sure you know that. It contains an ether, which is this hook right there. So 6-member ring containing an ether, but primarily you want to know it's a 6-member ring. Thromboxanes do 2 things. Actually, they do 1 thing we care about. They produce blood clotting. They produce blood clotting, which means that they are produced by platelets because platelets clot blood. Thromboxanes are produced by platelets. They cause blood clotting. Platelets, please know the name, are AKA thrombocytes, which is how you know they produce thromboxanes, because platelets are thrombocytes. They cause thrombus formation, blood clot formation, blood clotting, etc. If you see thromboxanes, platelets, thromboxanes, thrombos, thrombo, or blood clots. Any of those are synonymous for your test purposes. Thromboxanes are your major blood clotters. Right here's what we talked about already. We're going to go over it again, though, later. Non-steroidal and now. Non-steroidal anti-inflammatory drugs, NSAIDs, a.k.a. aspirin, ibuprofen, meclofenate, inhibit prostaglandin H2 synthase, a.k.a. COX, which catalyzes an early step in the synthesis path. So COX inhibitors stop inflammation. What else do they stop? What else does COX produce? It produces prostaglandins and, oh, true, it stops COX in the stomach, which allows you to get stomach ulcers, or might. But COX produces PGH2, and PGH2 is turned into prostaglandins and thromboxanes. What do thromboxanes do? Blood clot. So if you take aspirin and it stops the production of COX, what happens to blood clotting? There's no blood clotting, which is why you take aspirin as a blood thinner. What? Thins your blood. Exactly. So, a couple of reasons you'd be on aspirin. Anti-inflammatories, blood clotting, etc., etc. We're actually going to look at the specific mechanism of action of aspirin later. But the reason it works, the reason it thins your blood is because it stops the production of thromboxanes. The reason it decreases inflammation is because it stops the production of prostaglandins. We'll talk about that again. Oh, here we go. Cardioprotective effect of aspirin in myocardial infarction is mediated by TXA. In other words, it thins your blood, so the thromboxanes are what's important. However, PGI synthesis is also blocked by aspirin. However, aspirin also causes severe damage to the stomach. So, it also blocks PGI synthesis. It also blocks stomach stuff, etc., etc., etc. So it takes its toll on you. So now we're going to go to back to page 60. Back on thromboxanes. We've talked about this guy a little bit. Guess what? He has a 6-member ring containing an ether. 6-member ring. And he's got a double bond between carbons 13 and 14, and he's got an OH group at position 15. Those are your 3 structural characteristics of thromboxanes I want you to know. 6-member ring containing an ether, and double bond at 13, 14, OH group at position 15. Thromboxanes, responsible for blood clotting. Closely resemble prostaglandins, except the ring is composed of 5 carbon atoms and an oxygen. Thromboxanes are synthesized in platelets, aka thrombocytes, by thromboxane synthase, produces thromboxane A2, which is why it's thromboxane synthase. What was I going to say? Don't remember. Thromboxane synthase produces it. Oh, what's the precursor of thromboxanes? PGH2, prostaglandin H2, which is the first stable intermediate in the prostaglandin synthesis pathway. Nice job. On the top of page number 61, thromboxanes cause vasoconstriction, platelet aggregation, lymphocyte proliferation, and bronchoconstriction. They do the exact same thing as PGI, or PGF, sorry. They do the exact same thing as PGF, except instead of smooth muscle in the uterus, they affect platelets. They cause blood clotting. They cause lymphocyte migration. They cause the immune reaction to proliferate. They're causing stopping and damage repair at the area. So vasoconstriction, bronchoconstriction, platelet aggregation, and lymphocyte proliferation. They want you to repair. They want you to stop and shut down and get everything cooking there. They're rapidly inactivated, seconds to minutes half-life, TQ. They're inactivated by oxidizing the OH group at position 15, breaking the double bond at 13, or adding stuff to the ends or breaking the ring or any of that. If you change the structure of these guys, especially its key structural characteristics, you inactivate them. And it only takes seconds to minutes. Cool? All right, now key point right here. NSAIDs, non-steroidal anti-inflammatory drugs, inhibit COX. They are COX blockers. You need to know that aspirin is an irreversible inhibitor of COX, whereas ibuprofen, meclofenate, and acetaminophen are reversible inhibitors of COX. The reversible inhibitors of COX are competitive inhibitors. They just bind at the active site and stop COX from working. They don't let it get to any arachidonic acid. You've got to know the mechanism of action for aspirin, though, and we're going to go over that. So, ibuprofen, meclofenate, acetaminophen, et cetera, all of those are competitive inhibitors. They're reversible. You take them, the inflammation decreases, the half-life of the drug goes down, and it gets out of your system. Aspirin, though, is an irreversible inhibitor, and you got to know the mechanism of action right down here. I guarantee this will show up. Aspirin irreversibly inactivates COX by transferring an acetyl group to the serine hydroxyl group of the active COX enzyme. COX has a serine hydroxyl group available, and aspirin acetylates it, right down here. Here's your active COX with a serine and an OH group, because a serine is an alcoholic amino acid. Aspirin right here, acetylsalicylic acid, drops off an acetyl group, it's acetyl salicylic acid, becomes just salicylic acid, permanently adds an acetyl group to that COX. That COX is permanently inhibited, which is why they have you on aspirin as a long-term blood thinner, because it reduces your endogenous synthesis of COX. It stops you from clotting permanently, not just temporarily. You don't have to keep dosing up as often or every few hours. You can just take a small dose because you permanently decrease your enzyme. Do you think that causes other problems? Yes, aspirin is not great for you. And in fact, if you have to take an anti-inflammatory drug of any kind, and I'm not saying I don't, I have before and I probably will again, please don't take aspirin. Aspirin is a permanent enzyme damager. That enzyme hangs around until your body breaks it down, rebuilds it, and does whatever. So it throws off your homeostasis, et cetera, et cetera. At least a competitive inhibitor changes a little, but not quite as much. Anyone parked in the creek lot? Cool. Let's see where we're at. Let's try and blow through this stuff. Questions on that? Which one's irreversible? How does it irreversibly affect COX? An acetylates what? to the hydroxyl group at the serine of an active COX enzyme. So make sure you know that, please. It seems dumb, and it kind of stands out once you get to that question, talking about the mechanism of action. He will try and mess with you a little bit, but it's the one, I mean, please memorize it just in case he throws. But he will ask you that specific mechanism of action, please. Easy point. All right. We are done with thromboxanes and prostaglandins. We're now moving into the lipoxygenase pathway. That was the cyclooxygenase pathway, the COX pathway. The COX pathway produces prostaglandins and thromboxanes. PG-DI causes vasodilation, PG-DI being PGD, PGI, and PGE. Causes vasodilation, increase in CAMP levels, and decreasing immune function. PGI is AKA prostacyclin. It's produced by vascular endothelial cells. PGF causes the exact opposite, bronchoconstriction, vasoconstriction, smooth muscle constriction, and thromboxanes are produced by platelets, aka thrombocytes, causing blood clotting. That was a mouthful. We're going to go back and we're going to look at leukotrienes on page number 55 real quick. 55. Leukotrienes, what does a leukotriene mean? 3 double bonds in a white thing. So these have to do with white blood cells, kind of. They were first found in leukocytes, which means that we named them based on the leukocytes they were in. They're in lots of cells in the body, but they were first found in leukocytes. So this structural characteristic that you need to know is 3 double bonds. Please make sure you get that since it's called a leukotriene. If he says, which of these has 3 double bonds, it's the leukotriene. It's named for it. 3 conjugated double bonds. They're powerful biological signals. Fantastic. Leukotriene D4 is derived from yada yada. Causes contraction of the muscles lining the airways to the lungs. Causes contraction of the muscles lining the airways to the lungs. You've got to know that one. We're going to drill that into you. Bottom of page 55, contraction of the muscles lining the airways to the lungs. We're going to drill this again. I'm going to talk about it. Overproduction of leukotrienes causes asthmatic attacks because leukotrienes cause constriction of the airways to the lungs. What's that called when you constrict the airways to the lungs? Bronchoconstriction. So leukotrienes cause bronchoconstriction, and if you produce too many, you get asthmatic attacks. Leukotriene synthesis is one target of anti-asthmatic therapy. So leukotrienes are very responsible for asthmatic attacks. So, I can have a better picture than that. Okay. Yeah, right here, page number 62. Skipping back real quick at the top, it tells you what I told you. I didn't realize it had an actual statement on here. Acetaminophen, ibuprofen, et cetera, are competitive inhibitors of COX. They bind and then release normally. Competitive binding is reversible. We talked about that, but if you want where it's written in your notes, it's right there. Down here, we talked about the cyclooxygenase pathway. Now we're talking about the lipoxygenase pathway. The lipoxygenase pathway produces 2 things, leukotrienes and lipoxins. Lipoxygenase produces your 2 L's, leukotrienes and lipoxins. And hydroxyicoso, yeah, we'll talk about those. Not very long, because I can't pronounce them more than once. Leukotrienes, called leuko because they were first found in leukocytes. Structural characteristic, 3 conjugated double bonds. Leukotriene, 3 double bonds. Induces contraction of the muscles lining the airways to the lungs. Here it is again. And overproduction causes asthmatic attacks. It's like the 3rd time in 2 pages. Please make sure you highlight that. Please make sure you know that. Okay. We're going to show you this, but hydroperoxyeicosatetraenoic acids, HAPEATs, are produced. They're reduced to HETES, which, again, don't really need to know the name or the abbreviations. But you need to know that the major leukotrienes are all synthesized by the 5-lipoxygenase pathway. We're going to talk about 2 pathways. We're going to talk about 5-lipoxygenase and 15-lipoxygenase. All the leukotrienes, the major leukotrienes, come from the 5 pathway. The major leukotrienes are leukotriene A, B, C, D, and E. So the major leukotrienes are leukotrienes A, B, C, D, and E, and they're all produced by the 5-lipoxygenase pathway. Please don't say leucooxygenase pathway. It's lipoxygenase. A, B, C, D, and E. So looking on page number 63, you can see arachidonic acid up here going through 5 lipoxygenase, produces a 5-HPETE, HPETE. Did Dr. Sarkar pronounce that in class? HPETE. Have you been lectured on this yet? Yes. Speaking of which, what the hell is with your test schedule? Normally I have a week to do every chapter, and I only have 3 weeks with you until your next test, and I've got 4 chapters. Bullshit. Sorry. Sorry, podcast gods. Do we see what? Yeah. So 5 HPETs, then we produce 5 HTEEs, which doesn't even say, and then you produce leukotriene A, B, C, D, and E. Leukotriene synthesis. Bam. Easy. This chart is wonderful. It's probably not going to show up anywhere, but just so you see it. Arachidonic acid can go through the 5 lipoxygenase pathway and produce the 5 HPETs, then the 5 HEETs, or the 15 pathway producing the 15 HPETs and the 15 HEETs, or the one we don't even talk about at all is the 12 pathway producing 12 versions. 5 pathway produces the major leukotrienes. The major leukotrienes are leukotrienes A, B, C, D, and E, which produce bronchoconstriction. They're involved in asthmatic attacks. If you care about the name, I broke it down right there because I'm fancy. That's what that long, silly name means. You probably don't care, so I'm going to move on. But it's on vodcast now, so if you want to go back and read what this fancy name means, it's right here. All right. Moving on. Another one of those fancy lists for you. Leukotriene B increases immune function. In general, increases immune function. Saves you from memorizing all this random stuff. Increases vascular permeability, which is important for the migration of stuff from the blood to the cells. Causes edema, allows white blood cells to get out. It increases vascular permeability. Causes T-cell proliferation, part of your immune response. Causes leukocytes, part of your immune response. Interferons, interleukins, etc., etc. Leukotriene B increases immune function. Easy. Leukotriene B increases immune function. Leukotriene C and D increase bronchoconstriction, vascular permeability, and I don't even remember what INF is. This is the one that you want to know. Leukotrienes C and D are responsible for asthma. Leukotrienes B just increase immune function. Leukotrienes C and D are responsible for asthmatic attacks. They're responsible for asthmatic attacks. So, that's it for leukotrienes. They all come down the 5 lipoxygenase pathway, the major ones. They're produced from arachidonic acid. That's what they do, and that's all you got to know about them. We're going to talk a little bit more about the... let's talk about it now. So, what's our mantra for activation inhibition? Substrates activate. So what does that mean? Things that start the pathway activate it. So if you have a buildup of things that start the pathway, what happens to the pathway? It's activated. So what happens to an asthmatic kid that we give aspirin to, or acetaminophen or whatever? What does that aspirin and acetaminophen do? It what? No? Well, kind of, but how? You got to get me closer than that. Warm me up, come on. What is the mechanism of action, or what is the overall goal of an NSAID? Reduce inflammation, reduce thromboxanes, reduce platelets. Why? Because it decreases COX. So COX does what to what? It takes arachidonic acid and produces prostaglandins and thromboxanes. We want to stop that if we want to decrease inflammation or thin the blood. But what happens then? We have histamines, we have cytokines indicating tissue damage or allergic reaction. We produce free arachidonic acid by phospholipase A2 and phospholipase C, freeing arachidonic acid from the membrane. But then we take away one of the pathways that uses arachidonic acid. What happens? We get a buildup of arachidonic acid. A buildup of arachidonic acid drives the leukotriene pathway, the lipoxygenase pathway, because now we have a substrate building up that has nowhere to go. So if you have a kid, and this is theoretical, if you have a kid that you give aspirin to that's prone to asthmatic attacks or might be prone to asthmatic attacks, and you take out that COX pathway, they're still producing free arachidonic acid. That free arachidonic acid still has to go somewhere. You've taken one key guy out of the picture, so now it drives down the other pathway. It increases bronchoconstriction. Aspirin drives asthmatic attacks. Does that make sense? So interesting clinical correlate. I don't remember if he talks about it or we talked about it or we discussed it at some point. I don't know. But interesting, because leukotriene C and D increase bronchoconstriction, causing asthmatic attacks. It seems eerily quiet since the air conditioner went off. I want the air conditioner back. Okay. Now, the other guy produced by the lipoxygenase pathway is lipoxins. The lipoxygenase pathway produces leukotrienes and lipoxins and HPETEs, but those are just kind of whatever. We don't talk about them. Those are involved in the cytochrome system that we haven't talked about yet. Won't talk about. Lipoxins are produced by lipoxygenase. Now, specifically, please know this. The action of 15 lipoxygenase followed by 5 lipoxygenase produces a lipoxin. Remember, the leukotrienes are synthesized by 5 lipoxygenase. A lipoxin is made by going down the 15 lipoxygenase pathway and then the 5 lipoxygenase pathway in that order. Guaranteed, well, I don't guarantee anymore. I don't know how many of these will show up. But that's a great test question. It has shown up. I think we got asked it. And he put both of them on there. 15 then 5, 5 then 15, 5 and 5, and 15 and 15. You have to know it's 15 then 5. So if you take arachidonic acid and you put it through 15 lipoxygenase and then you put it through 5 lipoxygenase, which are 2 different enzymes, you get lipoxins. That's the only way you get lipoxins. If you do it any other way, you don't get a lipoxin. You get weird things. Leucoxins or something, whatever else. I don't know. 5 produces leukotrienes, 15 then 5 produces lipoxins. On page number 65, lipoxins do 2 things. They induce chemotaxis. Chemotaxis is the migration of white blood cells and other crud to that area that allows for inflammation or tissue repair and healing and all that stuff. And they stimulate superoxide anion formation in leukocytes. What this means, fancy words. These white blood cells, specifically I think neutrophils, I think, pick up oxidized LDL. We've studied LDL. They pick up oxidized LDL, or they pick it up and they oxidize it, and they create free radicals. And then the neutrophils chuck those free radicals are called superoxide anions, free radicals. They chuck those free radicals at bacteria. If your body cannot tackle the invader on its own, if it cannot use its own immune system, it doesn't have an antibody for it, if it cannot phagocytose it, the next step is for these guys to make free radicals and throw free radicals at the bacteria. Guess what free radicals do to bacteria? Bring it down. Guess what free radicals do to your tissue? Break it down. Cause cancer. Lots of stuff. But it's our own defense mechanism for like, well, we'd rather take out this small area than let whatever this is run rampant in our body. So your lipoxins induce chemotaxis, bring other stuff to the area, which helps in phagocytosis and repair, but they also bring out the big guns for the immune response. They bring out the, ooh, we can't deal with this in any other way. Okay, wow, we have covered this chapter in like an hour. That's impressive. Sorry. So, last but not least, mechanism of action of eicosanoids. They bind to membrane receptors, specifically the G protein coupled receptors, because they cause increases in CAMP. G protein coupled receptors, stimulatory G protein subunit, amplify signals. So remember there was 2 G protein subunits. There was a GI alpha and a GS alpha. The GI alpha turns off and decreases CAMP synthesis. The GS alpha, the stimulatory, turns on and increases CAMP synthesis. So there's 2 mechanisms of action. They can either increase or decrease CAMP. The one you got to know is leukotriene dye, prostaglandin dye, D-I-N-E, increase CAMP synthesis. They modulate the adenylyte cyclase enzyme and therefore cyclic AMP levels. And there are a couple calcium-dependent pathways, which is that IP3 pathway that we studied before and some other stuff. But primarily G-protein coupled pathways modulating CAMP in some way. That is eicosanoids. They are inflammatory mediators. They are blood clotters. They are vasoconstrictors, vasodilators, bronchoconstrictors, bronchodilators. They're involved in asthma, and they're taken down by NSAIDs. Any questions? How you guys doing? You want to take 5? It's cool. Take 5 minutes. I'm going to smoke you on this next section, too. I'm going to blow through it because I only have 3 weeks till your next test. I don't know how he's going to cover everything in that time because traditionally he moves slower than me. So we'll see what happens with your next test, but I don't want to be struggling towards the end of it and trying to get you guys caught up. This one and this next one are a very memorization kind of thing. I'm going to run you through some basic concepts in this next one, and we're going to give you a bunch of lists to take home and do on your own. So it'll be good to get it out of the way so you can start looking over it now. You've got a sheet with like 90% of everything on it. Yes, ma'am? No.
It started off in 95.
What?
We lost 5%.
Oh, sorry. I don't know. I talk out of... All right, I think we can smoke this chapter. Like I said, it's not a terribly difficult chapter. There's just a bunch of lists. We're talking about amino acid metabolism. I'm sorry, it's probably the most boring chapter we're going to be covering, just to get you all excited about this. We'll see what we can do about it. I don't have a lot of good jokes for this one, but maybe I'll come up with something. I'm sorry. I fail as a tutor. I can't tell a joke. So, we're talking about amino acid metabolism. What does this mean to you? This means that all of the amino acids you learned last try, you suddenly have to know again. So I hope you remember your amino acids and the story of alcoholic Alan and his escapades in drugs and the job market and sulfur and all that fun stuff. So that's what this is going to be dealing with. Specifically, not so much what the functional groups are and what they do and where they go, but what we do with the amino acids, how we make the amino acids and how we break them down, because you realize that we get these amino acids every time we eat. Anything you have that has protein in it is broken down into amino acids. We have to have a way to metabolize these amino acids. So we need to have something to do with them. So first things first, we're going to talk about nutritional importance overall. There are two conditions that you need to know. The two conditions are kwashiorkor and marasmus, both of which will be on boards. They were for me. Or both of which were on boards, and you got to know them for boards. You get two different types of diseases. The first one, kwashiorkor, is a protein deficiency. As you can see up here, poor sources of any of the nutritionally essential amino acids, which we're going to talk about, or present in inadequate amounts, can result in a protein deficiency. If these people aren't eating enough protein, they're deficient in protein. Now, this causes lots of different stuff. I don't think it's going to show up so much with him. You've seen this child on commercials. I hate to say it makes me sad, but it's a little African child with the swollen belly. That is kwashiorkor. They have plenty of grains, they have plenty of rice, they have plenty of whatever, but they're not getting enough protein. You can get enough protein from vegetable. It's just that they're eating too many polished, refined grains. They get, and you don't really have to know this for Sarkar, I don't think he asked this, the big one is going to be edema. When you get edema in your belly, you get that big swollen belly. It's called ascites, A-S-C-I-T-E-S, or swollen belly. You get decreased weight, decreased muscle mass, skin pigment changes. Anything that involves a protein in your body downregulates, slows down. As you can see, this is going to cause major problems. The edema primarily comes from the fact that protein is a major constituent of your blood. And you're going to talk about in physiology, I don't think you have yet, the osmotic pressure and oncotic gradient and all that stuff. Okay, basically the stuff in your blood, remember tonicity, hypertonic? The stuff in your blood allows your blood to draw fluid back in from the tissues. So when you get to a capillary and you perform fluid exchange, the only thing that keeps too much fluid from going out and enough coming back in is the pressure of stuff in your blood. The more stuff you have in your blood, the more fluid wants to come back in because proteins attract water. So if you don't have enough protein in your blood, you get to that capillary exchange bed and not enough fluid comes back in. Too much leaves and you can't get it back in because you've got no protein. You've got nothing to draw it back in. So you get edema, you get a swollen belly, ascites, all that stuff. Marasmus, on the other hand, is both a protein and a calorie deficiency. You are starving. You don't just lack protein, you lack... It's hilarious. I can't even believe you can do that. You lack protein and calories. This is going to cause basically the same problems: muscle mass, edema, decrease in adipose, dry skin, all that stuff. You need to know which one is caloric deficit and which one is protein deficit for Dr. Sarkar's test, for boards, all that stuff. Very important that you know that. Those will be definition questions, or one of those will be a definition question. So down here you see this nice little note. The amino acid metabolism involves the biosynthesis of amino acids and the degradation or breakdown. In other words, what the hell we do with amino acid. Of the 20 common amino acids, that are used in humans to synthesize proteins, 11 are synthesized and 9 are nutritionally essential. 9 are your essential amino acids. I need one of my own worksheets. Now, you haven't gotten there yet, but Dr. Sarkar has a great analogy. I suggest you use the amino acid metabolite, or the essential amino acid private Tim Hall, PBT Tim Hall. Much easier to remember. Private Tim Hall right there. Did I star these? So let's see. Of these ones that you need to remember, Private Tim Hall. You need to know phenylalanine especially because it has a proline. So what I'm doing is I'm going based on the letters, and I'm saying, of Private Tim Hall, which ones do I absolutely need to memorize? I need to memorize it's Private Tim Hall. And which ones can I ignore because there's only one V. So the only V is valine, and the only V is in Private Tim Hall, so you know which one it is. Phenylalanine you need to know because phenylalanine has to be differentiated from proline. Valine, you don't even really need to memorize because as long as you know Private Tim Hall and you know V is valine, it's the only V. Threonine and tryptophan are both over here, whereas tyrosine is over here. Now, it's not going to be too hard to remember, so tyrosine is non-essential, threonine and tryptophan are essential. Make sure you know that. You got to know all your nutritionally essentials. I'm just trying to make it a little easier on you. And we're going to teach you why tyrosine is not nutritionally essential. Isoleucine is the only I. Don't worry about it too much. Methionine is the only M. Don't worry about it too much as long as you know it's Tim Hall. Arginine. You need to be able to differentiate from alanine, asparagine, and aspartate. Arginine is the only nutritionally essential amino acid. In Chapter 7, we're going to learn the urea cycle. Arginine is produced heavily in the urea cycle, but we can't use it in anything else. We produce arginine in the urea cycle. It's used for the urea cycle. Therefore, arginine is a nutritionally essential amino acid for anything else that requires arginine, for protein synthesis, etc., etc. Arginine is made in the body in the urea cycle, though we just don't use it for we can't use it for anything else. And then lysine and leucine are the only L's, so you don't really need to memorize those. So, as long as you know your mnemonic, Private Tim Hall, there's like 6 of these you got to memorize. That's your first memorization for this test. It's not very bad. The other ones, don't memorize them. If you know the essentials, you know the non-essentials. He will ask you a couple of questions, I don't know how many, talking about which ones are nutritionally essential, which ones are nutritionally non-essential amino acids. These are great. Which one of these is not a nutritionally essential amino acid? Which one of these is not a nutritionally unessential amino acid? Because then you got to know more than just one. So, look for those on your tests. Questions on that? I'm not going to go over them much longer. It's just something you're going to spend a little time with. This section and eicosapentaenoic acids, eicosanoids, are a little memorizing intensive. Don't leave them for the end, please. Don't leave them like till the night before the test. Okay, so that's what your book says on page 67, your nutritionally essential amino acids are obtained from the diet. That's how all nutritionally essential things go. Arginine is an essential amino acid. Even though it's produced in the urea cycle, it's immediately degraded, so it's still nutritionally essential. Nutritionally non-essential amino acids we're going to be talking about for the first bit of this chapter. They're produced from glucose or intermediates of glycolysis of the TCA cycle. So anything we can produce them from. We can produce them from other amino acids or occasionally from ammonia, because the primary thing we need that we don't have very much of in terms of the other parts, the carbon skeleton, we can get from anywhere. But we need the nitrogen. So we have to get the ammonia, which usually floats around as ammonia, the nitrogen, from somewhere else. The only source of nitrogen in a human diet is a protein. So you have to eat protein to get protein either way. So, but if we have amino acids that we have plenty of, we can sometimes move them around, and that's what we're about to talk about. So, we have 3 transamination reactions that you must know. I think we talked about this last try. Transamination, ring a bell with the malate aspartate shuttle system. We're going to hit it again. You got to know those 3 again. You must know, I think you got to know it for this chapter, vitamin B6 is required for all transamination. Vitamin B6, aka pyridoxal phosphate, Pyridoxal phosphate, is the coenzyme that does transamination. It's going to be a TQ in Chapter 12 when we talk about water-soluble vitamins, but it also might show up on your transamination section. Vitamin B6 or pyridoxal phosphate does all your transaminations. Now, this is very easy. Let me draw somewhere. We take, who do we want to play with? Let's play with alanine. Okay, COOH, CNH3. You don't have to be able to draw this. I'm just demonstrating. What is that? What structure is that, first of all? Not like what specific thing is that, but is it a protein? Is it an amino acid? Is it a carb? It's an amino acid. What amino acid? Close. Glycine has an H here. Who's the main character of our stories? Alanine. That's alanine right there. So we take alanine, and we're going to combine it with any other alpha keto acid. So I'm just going to say this guy has a COOH here, and he's got 2 H's there, and he's got a CH3 there. That's an alpha keto acid. All it is an alpha ketone group on the terminal carbon. And what we're going to do is we're going to swap this NH3 to this guy right here. Now this is dumb because we're going to end up with the same thing, so I apologize. I should have a different scenario. There we go. Bam, there's an alpha keto acid that's different. This guy is not an amino acid. He has no amino group. He's just an alpha keto acid. But we're going to take this amino acid from alanine, and we're going to transfer it over there. What we end up with then is COOH, CH3, and this guy has H2 attached to him. This is pyruvate. And we end up with COOH, CH, H3. Valine. I know it's really complicated. The premise, no, I can't do that. The premise behind this is these amino groups are interchangeable. They are transaminated. We transfer the amino group. We take an amino acid and we combine it with an alpha keto acid, and we transfer the amino group, and the amino acid becomes an alpha keto acid, and the alpha keto acid becomes an amino acid. Because the only difference between an alpha keto acid and an amino acid is the amino group. In this case, alanine loses its amino group, it becomes pyruvate. Alanine has been transaminated to pyruvate. Whereas this random alpha keto acid picks up an amino group, and in this case, happens to become valine. This is transamination, the transfer of amino groups. There are 3 major transamination reactions you need to know. They're listed right here. I told you about them last time. Those 3 major transamination reactions are that you can combine, or you can transaminate pyruvate and alanine. And the reason you can do that is because the structure of pyruvate and the structure of alanine are identical except for the amino group. If you add an amino group to pyruvate at the right position, it becomes alanine. If you take an amino group from alanine at the right position, it becomes pyruvate. The only difference between those two. Now, if you'll notice, it has an amino acid plus an alpha keto acid becomes an alpha keto acid plus an amino acid, because there's only one nitrogen between one amino group between these pair, and it's going like this. That's the whole premise of transamination. They all require B6 because B6 serves as a carrier of the amino acid in between. You need to know these 3, though. These are 3 big TQs. They're 3 big ones on boards. If we want to make alanine, we transaminate pyruvate. All we need is an amino donor. Any other amino acid that can donate its amino group can turn pyruvate into alanine. Any amino acid. When that amino acid donates its amino group, turning pyruvate into alanine, it becomes an alpha keto acid. Have I made that clear as mud? Okay, questions? 3 you got to know. First one's the hardest, pyruvate to alanine. Back and forth all day, transamination. Other 2 rhyme. Not rhyme, they sound the same. Oxaloacetate and aspartate can be transaminated. The only difference between oxaloacetate and aspartate is the amino group in the right position. So if you transaminate oxaloacetate, you add an amino acid to it, or an amino group to it, it becomes aspartate. You take an amino group off aspartate, it becomes oxaloacetate. And alpha-ketoglutarate and glutamate. Take alpha-ketoglutarate and you add an amino group to it, it becomes glutamate. Take an amino group off glutamate, it becomes alpha-ketoglutarate. What's the common theme there? Those are all intermediates of glycolysis, right? You got 1 from glycolysis and 2 from the TCA cycle, which means that these intermediates of glycolysis can go towards protein synthesis, which means that if you're protein deficient, you're also going to decrease your intermediates of the citric acid cycle because you want to produce amino acids from them, at least these 3 amino acids. So these 3 amino acids, this is one of the ways they're produced. Pyruvate, oxaloacetate, alpha-ketoglutarate produce alanine, aspartate, glutamate. Don't not study those, please. All right, hang on. I know it's getting long. So that's on your little sheets right here. Transamination is reversible. It requires pyridoxal phosphate. pyruvate to alanine, oxaloacetate to aspartate, alpha-ketoglutarate to glutamate. Cool? Now the fun part. Here's where the flat-out memorization comes in. So I'm going to go through this real quick, and then we'll go back and we'll go through my notes. Alpha-ketoglutarate is the precursor of glutamate. Alpha-ketoglutarate is made from glutamate by transamination requiring vitamin B6, aka pyridoxal phosphate. Alpha-ketoglutarate is the precursor of glutamate. Then glutamate is the precursor of glutamine, proline, and arginine. Glutamate is the precursor of glutamate, proline, and arginine, which means that if we need glutamate, if we need proline, if we need arginine, if we need glutamine, proline, or arginine, we take alpha-ketoglutarate, we turn it into glutamate, and then we make it into the other three. These are the lists you're going to have to memorize. That glutamate produces glutamine, proline, and arginine. They can be interconverted. And like I said, I've got that on a little sheet that you can look over. So we'll go over that at the beginning. But at the very beginning, alpha-ketoglutarate to glutamate is transamination, and then glutamate can be converted to glutamine, proline, arginine. Glutamate produces glutamine, proline, and arginine. Bam. 3-phosphoglycerate, another intermediate of glycolysis, is the precursor of serine. Serine can be converted to cysteine and glycine. So another intermediate of glycolysis, 3-phosphoglycerate. 3-phosphoglycerate produces 3 small amino acids: serine, cysteine, glycine. 3-phosphoglycerate produces serine, cysteine, glycine. You can also produce glycine by this random method that probably won't show up. Oxaloacetate produces aspartate. You already know that one. Oxaloacetate produces aspartate. Well, then aspartate goes on to produce aspargine. Aspartate also produces methionine, lysine, threonine, and isoleucine. So if you want any of those, if you want methionine, lysine, threonine, or isoleucine, or aspargine, you produce aspartate, and you interchange them all. Again, what you need to know from this is the list. You need to know that aspartate produces those 5 amino acids, and I've got that in list form for you. Pyruvate, very small amino acid, produces alanine plus the branch chains. Pyruvate produces alanine plus the branch chains. Pyruvate produces alanine plus the branch chains. The combination of phosphoenolpyruvate and erythrose 4-phosphate, which comes from the pentose phosphate pathway, produces phenylalanine, tyrosine, and tryptophan. Both of those guys, phosphoenolpyruvate from glycolysis and erythrose 4-phosphate from pentose phosphate pathway, produce your aromatic amino acids. How do I know it's both of them? Because phosphoenolpyruvate is 3 carbons, and these guys are much more than 3 carbons. So the combination of phosphoenolpyruvate and erythrose 4-phosphate produces your 3 aromatic amino acids: tyrosine, tryptophan, and phenylalanine. And ribose 5-phosphate produces histidine. Super dry, I know. I love you guys. Hang with me. So if you look on your sheet, that's all made in nice little list form right here, which should save you from whatever you were just thinking about with a knife. Glycolysis intermediates, I broke it down by where they come from. Phosphoglycerate produces 3 small amino acids: glycine, serine, and cysteine. Cysteine can be turned into methionine. PEP, phosphoenolpyruvate, and erythrose 4-phosphate produce the 3 aromatics. Phosphoenolpyruvate has 3 names, produces your 3 aromatics: tyrosine, phenylalanine, and tryptophan. Pyruvate produces the branch chains plus alanine. Alpha-ketoglutarate produces proline, arginine, glutamate, and glutamine. You better know alpha-ketoglutarate produces glutamate. That's your big transamination, and then the other ones come from glutamate. Oxaloacetate produces aspartate. Aspartate produces methionine, lysine, aspargine, and threonine can produce isoleucine. So again, the list you got to memorize, they all stem from oxaloacetate or aspartate. And then ribose 5-phosphate produces histidine. Dry as heck, I'm sorry. Lovely list. Okay, now here's some minorly fun parts. We're going to deal with page number 69. These are cofactors or coenzymes that you got to know. Cofactors or coenzymes that you got to know. Pyridoxal phosphate, vitamin B6, which we've talked about already, is a derivative of pyridoxine, but it's pyridoxal phosphate, the active form, aka vitamin B6. It's required for all transaminations. Any transfer of an amino group, transamination requires B6. So your 3 big transaminations: oxaloacetate to aspartate, alpha-ketoglutarate to glutamate, and alanine to pyruvate. I reversed that one. Those all require vitamin B6 or pyridoxyphosphate. Now, you got to know this now, and you got to know this on test number 3, because this is the same question when we go through the vitamin section. Make sure you memorize this good, because it's going to be there. Questions on that one? We've hit that one kind of already. For example, transamination. Tetrahydrofolate, which is folate, which is vitamin B9, which is also going to be heavily tested on in exam number 3. Enzyme derived from folic acid, tetrahydrofolate. Tetrahydrofolate is the active form. Folic acid is the precursor. Accepts or donates 1-carbon groups. It's a 1-carbon transfer. So if we want to go from methionine to cysteine, and the only difference between cysteine, which has a terminal sulfhydryl group, and methionine attaches a 1-carbon group to the end of that. Tetrahydrofolate can donate 1-carbon groups. The big one he's going to use here, and that was a bad example because that's not what directly does that, and we'll study that pathway later. Here's your better example. Serine and glycine. Difference between serine and glycine, aside from the fact that serine has an OH group, is that serine has 1 extra carbon in the side chain. So if we want to turn serine into glycine, tetrahydrofolate can take the carbon off there. If we want to turn glycine into serine, tetrahydrofolate can add a carbon to it. So it participates in 1-carbon transfers. Your TQ example for tetrahydrofolate would be serine to glycine. So if he asks you, what coenzyme is responsible for the conversion of oxaloacetate to aspartate? Conversion of oxaloacetate to aspartate. Vitamin B6, aka pyridoxyphosphate. What if he asks you what cofactor or coenzyme or vitamin is required for the conversion of serine to glycine? Tetrahydrofolate or folic acid. He won't ask you B9, but that's for your information. Folate is vitamin B9. Questions on those two? Last one is the reason that tyrosine is not an essential amino acid. Because we have a vitamin, oh, I forgot to mention that. Tetrahydrofolate is aka THF. Or I think he also refers to it, let me write this one in here, as FH4. FH4, folate with 4 hydrogens on it. FH4 or THF, tetrahydrofolate or FH4. The reason I want you to know that is because tetrahydrobiopterin is not tetrahydrofolate, is BH4. BH4, biopterin with 4 hydros on it, hydroxys. That's why they're tetrahydro, that's why they're abbreviated like that. This does a very specific thing. Tetrahydrofolate transfers 1-carbon groups. Tetrahydrobiopterin hydroxylates rings. It adds a hydroxyl group to a ring structure. In other words, if you take phenylalanine and you add a hydroxyl group to it, it becomes tyrosine. It hydroxylates any ring structures. Phenylalanine to tyrosine is your big one. So the reason that tyrosine isn't an essential amino acid is because phenylalanine is an essential amino acid, and we have an enzyme that hydroxylates it from phenylalanine to tyrosine. So you don't need to eat tyrosine. You can make tyrosine as long as you're eating phenylalanine. Cool? So ring hydroxylation or phenylalanine to tyrosine is your TQ example. Make sure you know those 3 examples, or I'm sorry, there's 3 examples for pyridoxyphosphate, 3 transaminations. 1 example for tetrahydrofolate, 1 example for tetrahydrobiopterin. Also know it's just, in general, ring hydroxylation, 1-carbon transfer and transamination. Okay. That's on here as well, somewhere. Your active forms, pyridoxyphosphate. Your active form is pyridoxyphosphate. Your inactive form is pyridoxine or vitamin B6. It does transamination. Tetrahydrofolate, aka THF or FH4, is folic acid or vitamin B9, accepts and donates 1-carbon groups. And tetrahydrobiopterin is ring hydroxylation. It's all on your little sheet. Okay, we've talked about the synthesis of amino acids. We've talked about the nutritional essential amino acids. Now we're going to talk about the degradation or breakdown of amino acids. There are 2 types of amino acids. Well, there are 3. There are glucogenic amino acids, and there are ketogenic amino acids. And then there are those amino acids that are both glucogenic and ketogenic. Glucogenic amino acids break down and turn into glucose intermediates. Ketogenic amino acids break down and turn into ketone bodies. That's all it's saying. Glucogenic break down and turn into intermediates of glucose. Ketogenic amino acids break down and turn into ketone bodies. So, all this stuff it says at the bottom, 70, I'm going to go over on my little chart. I'm not going to go over it in your book because it says the same thing. What I want you to flip to is I want you to flip to page number 72. Because page number 71 says all the same stuff, has some wonderful little charts for you to look at. Page number 72, amino acids that are both ketogenic and glucogenic. They break down into both ketone bodies and glucose intermediates. Tryptophan, phenylalanine, and tyrosine, and isoleucine and threonine. Your three aromatics, isoleucine and threonine. Now, isoleucine and threonine like to go together. You can consider them your little stepbrothers. Because isoleucine and threonine both are synthesized from that common intermediate back at the beginning. So if we look back here, where is that? Oh, right here. Down under oxaloacetate, threonine and isoleucine are together right there. Threonine and isoleucine are together right here. So, not that that helps you that much, but just a little bit. So these amino acids are both ketogenic and glucogenic. Tryptophan, isoleucine, and threonine produce acetyl-CoA. That's going to be on your sheet. And phenylalanine and tyrosine produce acetoacetate. Acetyl-CoA isn't a true ketone body, but it will be turned into ketone bodies. Again, your ketogenic, glucogenic amino acids are tryptophan, phenylalanine, tyrosine, isoleucine, and threonine. Easier, your strictly ketogenic amino acids, lysine, leucine. Ketogenic amino acids, lysine and leucine, your two L's, lysine, leucine. Which means that every other amino acid that we didn't talk about is purely glucogenic. So, you need to memorize 7. Lysine, leucine, ketogenic. Aromatics, isoleucine and threonine, ketogenic, glucogenic, the rest are glucogenic. He tends to ask about these 7, though, not so much the purely glucogenic amino acids. Now, okay, down here on your sheet, near the bottom, the degradation of amino acids. Amino acids are broken down into 2 skeletons. They're broken down into a carbon skeleton and a nitrogen skeleton. The nitrogen skeleton is the amino group. All that the nitrogen skeleton is the amino group. In Chapter 7, the next chapter, we're going to talk about what we do with the amino group. We run it through the urea cycle because we have to excrete nitrogen as a waste product. We get all our nitrogen from protein sources, and we have to be able to excrete nitrogen as a waste product. So the nitrogen skeleton is broken down in the urea cycle. We'll talk about that next chapter. The rest of it is the carbon skeleton. Everything but the amino group is the carbon skeleton. And that carbon skeleton can be broken down into ketone bodies, glucose or ketone bodies, or just glucose. So here's your list right down here. Ketogenic amino acids, lysine and leucine. Glucogenic and glucoketogenic amino acids, and you might refer to it like that, keto-gluco or gluco-keto. Aromatics, threonine and isoleucine, and glucogenic is everything else. So please note that both glucogenic and ketogenic can be summed up by glucoketogenic or keto-glucogenic, however he chooses to write it. All right. Now, this apparently didn't make it on the back of the sheet. Thank you, printer. What? Yeah, that's what I mean. It formatted wrong. I blame the printer, not my ineptitude, just so we're aware. Here's your list for stuff we just skipped, and I'm going to breeze through this because you're as bored with it as I am. I'm sorry. This is the breakdown of amino acids. We talked about the synthesis of amino acids. Now, the reason I highlighted it like this, and let me make sure before I tell you. Yeah, okay. The ones you need to memorize that are different are the italicized ones. Cysteine, serine, and glycine are produced by pyruvate. Guess what? When they're broken down, they break back down into pyruvate. So pyruvate produces cysteine, serine, glycine, and cysteine, serine, glycine, when they're broken down, produce pyruvate. But also alanine and threonine, when they're broken down, produce pyruvate. So some of these are the exact reverse of synthesis, some of them are. I tried to make you able to study it a little bit more by being able to reverse these. They're new. So pyruvate can produce cysteine, serine, and glycine. If we're making amino acids from pyruvate, we can make cysteine, serine, glycine. But if we're breaking cysteine, serine, glycine down, we can make pyruvate. So they work, it's a reversible reaction. The only thing is, pyruvate cannot produce alanine or threonine, but when alanine and threonine break down, they produce pyruvate. So the ones I italicized are the ones that aren't exact reversals from synthesis pathways. This is the degradation of amino acids. Those 5 break down to pyruvate. Glutamate, glutamine, proline, arginine, and histidine produce alpha-ketoglutarate. Again, every one of those is an exact reversal except for histidine, because alpha-ketoglutarate produces glutamate, glutamine, proline, arginine, does not produce histidine. So you got to memorize histidine. Aspartate and aspargine produce oxaloacetate. Those should be pretty obvious because oxaloacetate produces aspartate, aspartate produces aspargine. Isoleucine, methionine, and valine produce succinyl-CoA. That one's brand new. Isoleucine, methionine, and valine produce succinyl-CoA. And then your mixed ones. Isoleucine, threonine, lysine, and leucine produce acetate and acetoacetate. Tryptophan produces both alanine and acetoacetate. Remember, tryptophan's huge. When it breaks down, it gives you both alanine and acetoacetate, both a glucogenic intermediate and a ketogenic intermediate. And this is glucogenic because it can go to pyruvate. So remember, tryptophan is one of your glucoketogenic amino acids. And phenylalanine and tyrosine produce fumarate and acetoacetate. Those are horrible. I'm sorry. They're lists. I can't make any fun with lists. Well, not biochem lists. There are other lists that can be good. Make your own list. All right. So, last but not least, and then you are freaking out of here, is we got a couple disease states to cover. Do you have any questions over the meat of this chapter, which is boring as hell? Sorry, Dr. Sarkar. Please don't listen to the vodcast. I'm going to get in trouble. I'm not going to get in trouble, but he's going to be like, damn it, Paul, you're not allowed to be my tutor anymore. You make this sound so dull. So, defects in amino acid metabolism. We've got like 5 or 6 of these to cover, and like I said, I'll get you out of here. Defects in glycine metabolism. If you cannot metabolize glycine, the enzyme that is deficient is glycine transaminase. What do you think glycine transaminase does? Bam, transaminates glycine. What cofactor does it require? B6, aka pyridoxyphosphate. Nice job. If you cannot transaminate glycine by glycine transamylase, you accumulate glycoxylate, and you get primary oxaluria, which causes kidney stones. Primary oxaluria from glycoxylate. Fun memorization, too. Here's one that you're going to hit hard in nutrition. Next. Try 4. Next try. You guys are 3? You guys are 2? Oh, you're so young. Defective methionine degradation causes hyperhomocystinemia. You're going to learn, we'll talk about it in Chapter 8. Ooh, that'll be on this test. So we'll hit this really hard. This will make a lot more sense by the time we hit Chapter 8. Methionine and homocysteine intraconvert. They're intimately connected. If you cannot degrade methionine, you build up homocysteine. And you'll understand that by the time we get to Chapter 8, so don't worry about it too much. If you cannot degrade methionine, you build up homocysteine. The disease is homocystinemia. The enzyme that's deficient is cystathione beta synthase. Cystathione beta synthase, and you get cardiovascular problems. Homocysteine is an independent risk factor for vascular and heart problems. So, methionine degradation, deficient cystathione causes homocystinemia, which is a buildup of homocysteine, which is a vascular risk factor. Now some fun ones. These are dangerous. You got to pick these ones up in kids early. Defective phenylalanine degradation leads to phenylketonuria. Have you guys ever heard of phenylketonuria? Aha. This is a pack of Trident. I know, shut up, I eat Trident. Phenylketonurics contains phenylalanine. This is a very deadly disease that very damaging disease that if you cannot degrade phenylalanine, you need to pick it up early. because this is kind of prevalent, all food that contains phenylalanine must be labeled as such containing phenylalanine. So if they pick this up early enough in kids, they can live a totally healthy, normal life as long as they don't eat phenylalanine. But if you eat phenylalanine, you get problems. I don't know how they work around the fact that it's an essential amino acid, but the defective enzyme is phenylalanine hydroxylase. What does phenylalanine hydroxylase do with it? Hydroxylates it. What structure does phenylalanine have? It's aromatic, which means it's a ring. What vitamin does ring hydroxylation? BH4. And phenylalanine turns into tyrosine. So if you cannot degrade phenylalanine to tyrosine by hydroxylating it with BH4, you get a buildup of phenylalanine. It's called phenylketonuria because it's got ketogenic properties. High levels of phenylalanine and phenylpyruvate, which is a phenylketone, lead to neonatal vomiting and mental retardation. This happens very early. By the time hopefully they pick it up early enough. But if they don't pick it up early enough, you have permanent brain damage from a buildup of phenylalanine and phenylketones in a baby. So phenylketonuria, look on your packs of gum. I don't think cinnamon gum has phenyl. Yeah, it doesn't need the sweetener from the phenylalanine. I think that's the one. It'll say it somewhere on there. I heard that one.
It says it contains phenylalanine.
Okay. Some of them. Maybe it was Trident didn't have it or something like that. So if you're phenylketonuric. Now this one's a little better. We can have a little bit of fun with this. This is alkaptonuria. Spelled two different ways, whether it's with a C or a K. I don't care how you spell it. Alkaptonuria. This is a problem with phenylalanine and tyrosine degradation. The enzyme deficiency is homogenticid oxidase. Homogenticid oxidase causes this problem. An accumulation of homogenticid oxidase causes black urine. So what I want you to, dark pigment in the urine and late developing degenerative arthritis. Great. Arthritis and my pee is black. What I want you to think about with alkaptonuria is the sea captain with the big old black beard. And he's out there on the ocean, and he's got this defect, and every time he pees, it's black, and he's a famous pirate because of it. Alkaptonuria causes black urine. The defect is homogenticid oxidase. You'll study this a little bit later in one of your lab diagnosis, I think. And the last one, which you're very excited about, defective branch-chain amino acid metabolism. What amino acids can we not metabolize? Branch chains, which ones are those? Nope. Valine, leucine, and isoleucine. Valine, leucine, and isoleucine. You might want to review some of those just in case he decides to throw a left curveball saying, you cannot metabolize valine. And you're like, oh crap, I didn't study that. Well, you did. You just didn't know that valine was a branch chain amino acid. Or maybe write that in right now. Valine, leucine, isoleucine. Valine, leucine, and isoleucine. Or maybe, it says it right there. Valine, leucine, isoleucine. Bam! Top of the paragraph right under defective branch chain amino. Oh. It doesn't say it on there? I failed. I'm sorry. Branch chain amino acid, The enzyme defect is branch chain aminotransferase. What does it do? It aminotransferases. And you guys get by now that the whole point of this aminotransferase, and we're going to deal with this in Chapter 7, so you're going to get drilled on it there, is that we need to get rid of the nitrogen skeleton. Once we get the nitrogen off, which we use the urea cycle for, we have this carbon skeleton that we can break down. If we can't get the nitrogen off, if we don't have the right amino acid transaminase, we have an amino acid. We can't use it for anything else because it's got a nitrogen stuck on it. So defective branch chain amino acid transaminase or branch chain alpha keto acid dehydrogenase. So first you transaminate an amino acid, it becomes an alpha keto acid. Then you take your alpha keto acid and you dehydrogenase it and becomes something completely different. But both of those result in a buildup of branch chain amino acids, valine, leucine, and isoleucine, accumulate in the blood and are distributed in the urine. It causes maple syrup urine disease. Anyone want to take a guess as to why it's called that and who the hell figured it out? I think it smells like maple syrup. It might taste like maple syrup. But these are traditionally sweet amino acids. When they're found in the urine, it gives it a characteristic smell and theoretically taste. Because believe it or not, before we had urinalysis, that's how they used to urinalysis. is they would literally smell and taste the urine, hence why diabetes mellitus, sweet, yep, okay. Maple syrup urine disease, symptoms, vomiting, convulsions, abnormal brain development, mental retardation, poor myelination, and early death. Sounds like fun. All of these are on the bottom of your sheet. As someone so lovingly pointed out, I failed and didn't write your branched chain amino acids right down here, but you have all 5 of those. I'm not going to go over them again. It has them nicely listed as the enzyme, what accumulates, what the disease is, what the symptoms are, and any other fun stuff. It's a memorization chapter. I love you guys. Thank you for drilling through 2 chapters with me. Luckily, you have the vodcast, so you can listen to these wonderful, enthralling lectures again if you so desire. Feel free to ask me any questions, and thank you for being here. Yeah.